everybody. That was that's a very hard talk to follow. I feel like everyone needs a little bit of a stretch first too. So I guess one of the most striking things about the natural world is the richness of colour, at least at the level that we can see. It's always inspired human art. It enables us to communicate, and many animals as well, of course. I mean, just imagine if all of the slides that you'd seen over this series of talks had been in black and white, the information would have been much harder to process. So, in addition to having all of these functions, it of course has inspired a lot of scientific inquiry. And I guess if I were to describe what I do, my research, it would be to say that I try and explain that aspect of biodiversity, so the colours in nature, from a mechanistic and functional and evolutionary perspective. Now, when we think about colourful animals, we often think about birds. But in fact, birds are an exception, if you like, because they produce these vivid colours in essentially dead tissue, in feathers. Whereas the great majority of vertebrates, more than 75% of them, in fact, so fish, amphibians, reptiles, produce vivid colours in skin, in living tissue. And these colours can be incredibly vivid. So how do they do it? Well, Atanasius already introduced these pigment cells, but essentially they occur in a layered system. So at the deepest layer we have melanophores containing these organelles of, with melanin pigment in them. And these cells are often involved in really rapid colour change over the scale of seconds. And that's because these organelles can migrate deep towards the cell centre and reveal the colours of the layers above, or they can migrate towards the skin surface, and the animal darkens. In this second layer here, we have iridophores, and they contain these reflective platelets. And when those platelets are very regularly shaped and arranged in an ordered way, they reflect specific wavelengths of light. So colours like blues and purples and iridescent colours. And then the icing on the cake here is these xanthophore cells that contain red and yellow pigments, often carotenoids and pteridines. And they essentially act as a coloured filter. So you can imagine that if these cells produce blue overlain by a yellow filter, the animal appears green. So with a relatively simple layered system like this, you can produce that extraordinary diversity of colours. And it turns out that, this is a picture, of a single scale, and at the microscopic level it's almost like an impressionist painting. You can see the arms of the melanophores there. Now it turns out that mammals and birds have lost this layered system. This layered system is ancestral. And mammals and birds only have a single type of pigment cell. You might be wondering how birds produce all those colours then with a single type of pigment cells, the melanocytes. And it's because they can deposit pigments like carotenoids directly into growing feathers, and they can produce structural colour just by having a very ordered arrangement of melanosomes. But nevertheless, they've still got only one type of pigment cell. And because it's this pigment cell that's found in mammals, including humans, we know an awful lot about them, about the melanophores, but we know a lot less about these other pigment cell types. And what we know comes from a very small number of model organisms, things like the zebrafish, and those model organisms don't include any animals from a major vertebrate lineage, which is the reptiles. In addition to, not knowing much about these pigment cell types, we actually know remarkably little about the pigments within them. And that's because, that actually is surprising, because one of these groups of pigments are pretty ubiquitous, and they're not limited to vertebrates. They're probably the most abundant group of pigments in nature. They're often found in invertebrates as well. So it's surprising how poorly known they are, even though they're so widespread. And they're also quite intriguing for other reasons. So within the xanthophores, we know that they are found in these organelles with these characteristic concentric lamellae, but they might be found in the other pigment cell types as well. And what's intriguing about them is that they not only include coloured pigments like Drossopteran, which is named for giving Drosophila its red eyes, but they also include a large number of colourless compounds that are found in really large quantities in the skin of many of these vertebrates, and in quantities much larger than you'd expect from their known kind of metabolic roles. So as I mentioned, there are these coloured pteridines, colourless pteridines. I've just listed 4 here, but there are quite a few of them, and in particular xanthopteran is intriguing because it can form highly reflective crystals, and we know it forms these reflective crystals in the eyes of birds and some crustaceans, and that was shown by a collaborator in our project, Ben Palmer. So, and I added this just before lunch for the chemists amongst you. And so, given all this, basically what motivated our project was to find the missing pieces of the puzzle about the composition and the molecular control and development of xanthophores, and we hypothesized that colourless pteridine pigments in particular that are found in high quantities could actually contribute to light reflection rather than absorption like typical pigments. And you've already heard Athanasia talk about this second goal, which was looking at the development and control of these xanthophores. So this was our team. You've heard from Athanasia this morning and the work of her postdoc. The work that I'm going to present today is mainly the work by postdoc Adrian Lutz, who's a biochemist, and he did some very detailed quantification in molecules per skin weight of the pigments in a wide range of reptiles, which I'll show you in a second. So these are the species that we were working on. So there were a range of lizard species, and then Athanasia talked about these colonies, amazing resource actually, of these corn snakes that have various colour morphs, and that means they've got mutations that impact their pigment cells. And so, of course, if we understand the causes and the consequences of those mutations, it's a very powerful system. Now, one of these lizards, actually 2 of them, these 2, are found in India. And one of them we chose specifically. So I should say we chose them because we know they contain pteridine and carotenoid pigments in their skin. But one of them is actually remarkable, this particular species. It's found in India, and normally it's this drab colour, this the female, this the male, fairly drably coloured. But then in the breeding season, when males court females, they switch on these really vivid colours, bright red back and black on the sides. And they only do it, they can do this very quickly, they change in a matter of seconds, and they only do it when they're courting females. Whereas when they're defending territories against males, the back turns from red to bright yellow and the sides turn from black to red. So I think I've worked on chameleons for many years, and I think these lizards beat chameleons because chameleons basically have the same repertoire of colours when they're displaying to males and females, whereas these turn entirely different colours. And so the reason that we looked at these is because we thought the xanthophores must be involved. If they switch from bright red to bright yellow, that would be an entirely new mechanism of colour change in reptiles. So that's a little bit of a side story. I don't have time to go through data for this today, but happy to talk to people about what we've found there. So far I've only mentioned carotenoids and pteridines as red to yellow pigments, but what Atenasia observed in her corn snakes were that there was one morph where there were no xanthophores, so it's a mutation where they've got no xanthophores, but they have yellow patches. And she thought, well, look, if it's not carotenoids or pteridines, what can it be? And it turned out that there were some very old reports of riboflavin in the skin of snakes from the 1960s, and actually a paper in Nature in 1954. So this is the entire paper. They couldn't show that it was involved in skin colour, just that it was found in skin. And so almost as an afterthought, Adrian quantified riboflavin in a separate extraction. So it's not a pteridine, not a carotenoid, different solvent and different extraction. And what we found actually surprised us. So we found that riboflavin was found in high quantities in all of our samples. That in itself isn't surprising. But those concentrations differed within individuals in different skin samples. So in this lizard, for example, you've got higher quantities of riboflavin in the kind of yellowy brown dorsal part and the yellow stripe than in the cream ventral area. And with our very cool Indian agamid, you've got higher amounts in the part that goes from yellow to red than in the part that just goes from red to black, so no yellow. And it was even more marked in snakes. So this is a morph where you get higher amounts of riboflavin in the richer orange patches than in the background patches, and higher amounts in wild type yellow skin than in the kind of red brick patches. And so this was kind of intriguing because riboflavin, like carotenoids, is obtained from the diet. It's very important metabolically. It's vitamin B2. Very important metabolically for cell growth. It's an antioxidant. And so here it suggests that as well as being important for healthy skin, and in humans actually it's a dietary supplement, as well as reptiles, it actually contributes to skin colour, and that actually has quite a lot of important evolutionary and ecological implications that fascinate me. So that was unexpected. We also showed that I guess the colour of the skin was very strongly correlated with the concentrations of coloured pteridine pigments. This in itself is not surprising, but the strength of those correlations was surprising, and it's probably because of the precision of Adrian's work. That was not the case for carotenoids. There's a correlation, but it's very, very weak, and they're found in much smaller quantities. So what about the mysterious colourless pteridines? Well, it turned out that our hypothesis was wrong. If they contributed to light reflection, we would expect a positive correlation between concentrations of these pigments and brightness, and instead we see the opposite. And this can just arise as a secondary consequence of being correlated with concentrations of the coloured pteridines, because you see this same negative correlation with brightness. Ben's group did some ultrastructural work, so this shows the xanthophores and iridophores, and actually found that the only evidence of any crystalline material was in the iridophores, not in the xanthophores. And then when we looked, or he looked at the traces, only found evidence of guanine, which we expected in these iridophore crystals, not of isoxanthopterin. So there's absolutely no evidence that these colourless pteridines actually form crystalline material in these skin cells and contribute to brightness. So where are they and what do they do? We do know a little bit about pteridine biosynthesis, but only from zebrafish and fruit flies. And I guess when we look at pigment correlations, it gives some clues, but it doesn't mesh entirely. So there's a lot we don't know actually about the biosynthesis pathway. So there are strong correlations between some pigments which could indicate that they're part of a common pathway, but actually that can't explain a lot of the patterns. We can get more clues by looking at mutations in the corn snake morphs. So this shows pigment concentrations. I'm sorry, it's a busy slide, but you can see, for example, in this cinder morph of the corn snake, it has no isoxanthopterin, and Atenase is identified the specific mutation involved. So we can start getting clues. Sorry, and I should say this snake has very few xanthophores, and their organelles are heavily impacted. So it gives us clues that it may be limited, I guess, to the xanthophores. And so overall, I guess what I wanted to do in this talk is to tell a story about a hypothesis that we had that motivated our Human Frontiers project and turned out to be wrong. We had good reason to believe that it might be true, but it wasn't. And in the process of, I guess, doing this research, we found some discoveries that we weren't anticipated and potentially more interesting. I think the riboflavin story has pretty wide implications. It may well be very abundant in many vertebrates, not just reptiles, and contribute to skin coloration. There were some findings that were more expected, such as the correlation between skin colour and pteridine concentrations, and there's a lot that we're pursuing at the moment to try and understand more about the location and molecular control of these pteridine pigments and xanthophore cells. And then an interesting offshoot of all of this, which actually wasn't part of the original project idea, is a potentially new mechanism of, actually it would be a new mechanism, it is a new mechanism of colour change in reptiles involving motile organelles containing red drosopterin in these xanthophores. So that'll be for the next HFSP meeting. Thank you.